Welcome to Popping the Science Bubble, everyone. This is a collaboration between the Berkeley Public Library and UC Berkeley, and it is a monthly seminar series that aims to share new research findings from graduate students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have two speakers at each seminar who will talk about their current research on a topic that they study or that they just find really interesting. The organizers are three graduate students at UC Berkeley, Jenna, Madison, and Oksana. If you're interested in checking out their past seminars, you can find them on YouTube. And we encourage you to check Facebook and to check the library calendars to see future events. Events happen on the third Tuesday of the month at 5:30 p.m. All right, and I'll let the team introduce tonight's speakers.
Hi, so we are Popping the Science Bubble. The organizers here are me, Madison, Jenna, and Oksana. We are current grad students at UC Berkeley. And we're real excited that today our first speaker will be Mattina Alonge. So Mattina grew up in New York and completed her bachelor's at Stony Brook in biochemistry. And then after undergrad, did a master's degree studying fish physiology. And then of course took 4 years off of schooling, working in the science and service industry, and now works on her PhD in exploring the impact of environmental challenges on reproduction in birds and bats. And she says at Berkeley explicitly, or especially, she loves the opportunity to spend time outdoors and has been able to go to Madagascar for 5 weeks to do some really cool research. So we're really excited to have her here to talk about this and introduce us to the world of bird parenthood. So take it away, Mattina.
Thanks, Madison. Yeah, I do study bats as well, but tonight is going to be bird focused. So if you're interested in bats, you can always reach out to me or contact me. I'm happy to chat about bats too. So yeah, tonight I'm going to be talking about how birds juggle taking care of themselves versus taking care of their offspring and babies. And we'll be thinking about this specifically in terms of zebra finches, which I will introduce later. So you probably have heard this in your life at some point, that balance is everything. And juggling responsibilities is something that we are probably all really familiar with. So whether it's finances, family, employment, or whatever you might do for income, self-care and mental health, or physical well-being, balancing all of that is extremely difficult. And animals, I used a bird here because we'll be talking about birds today, but all animals also juggle a variety of responsibilities or physiological or biological processes. So these might be things like growth or maintaining their body, parental care, which we'll talk about more later, things like migration or traveling from different locations to new locations, foraging for food and other resources, or maintaining their immune systems and immune function. So again, balance is important, but for non-human animals, it's not just a matter of mental health, it's a matter of survival. So if they don't manage these things properly and budget these things in the correct way at the correct time, then it often might result in some bad news for that animal. So making trade-offs are a part of our lives. We're most familiar with this concept in terms of money. So most of us here, I imagine, don't have an infinite supply of money growing on trees. That would be nice, but not usually the case. So money is a finite thing that we have to deal with and budget. And so maybe we decide that we want a super fancy Maserati, but that might come at the expense of having a really, really tiny house. Of course, this could go the opposite way, right? We want a really fancy house, but maybe that means we can't afford a super fancy car, so we have a more modest way of getting around. But Trade-offs don't just refer to finances. We can also think about them in terms of time. So time is a valuable resource, and there isn't an infinite amount of it. So we also budget time. And in this case, I'm really glad that you're here and you made the choice to invest your time in attending a Popping the Science Bubble talk. And what you may not have considered when you did that is that came at the expense of holding a socially distanced, properly masked party with your friends. So thanks for being here and tuning in and listening to the talks tonight. So in nature, resources available to animals are also not infinite. And we usually think about this in terms of time as well, but energy in addition. And classically, the two main places that animals have to budget these resources across are between behaviors or processes that support reproduction, or behaviors and processes that support maintaining their body or survival. And so in this case, I'm showing as an example a greater investment in maintaining their body condition, and that might come at the expense of reproducing in a given year. So maybe they forego having any offspring one year because they have to actually focus on staying alive. Of course, this could be flipped the opposite way, where maybe an investment in reproducing comes at the expense of maybe maintaining their body in peak physical condition. Again, any parents in the audience probably understand that. So in the animal kingdom, reproductive things that we might measure are the number of offspring an individual might produce, the number of mates it might try to court or attract, investment in the size of eggs in birds, or resources or nutrients into those eggs. And for body maintenance, examples of this in the animal kingdom would be things like growth rate, adult body size, or immune function or immunity. And biologists have a fancy name for this that I thought I'd throw in here. It's called life history theory, and this is a really central tenet to that theory. For the purpose of today, we're actually going to be focusing just on behavior. So we're going to be talking about parental care as a measure of reproductive investment. And for body maintenance, we're going to be looking at sickness behavior, which I'll talk about in a minute, but you can consider this self-care. So I kept this slide simple. Parental care is energetically demanding and time consuming. Enough said. I don't think I have to defend that with any fancy explanations. So we'll just keep going. The important thing I do want to talk about a little bit here is that animals get sick too. So we deal with colds, COVID, but animals experience a lot of different types of infections as well, whether they're bacterial or fungal or viral. And there's often a negative relationship between any sort of reproductive activity and immune function across many different animal species, not just birds. I want you to look at here on this graph as a really simple example. This is a bird experiment, and they increased by this +2 indication the number of babies that birds had. And when they did that, the more babies that these birds had resulted in a larger infection. So infection degree is basically plotted upwards. And so this increased bar shows a more intense infection for birds that had more babies. We also know that the exposure to something that makes animals sick, or pathogens, cause a really well-defined response. And this includes this sickness behavior or self-care. So that's defined as a decrease in locomotor activity or moving around, declines in appetite or feeding and drinking, increased time spent hanging out, resting, decreases in body condition, and changes in body temperature. And all of these things probably sound very familiar because these are the same types of symptoms and behaviors that we exhibit when we feel kind of crappy. So lastly, social environment can also influence what animals do when they're sick. And so here, this was actually an experiment conducted in zebra finches, the animals I'll talk about in more detail. And what they did is they used something called LPS, which activates the immune system and makes the animals think that they're sick. And these birds, zebra finches, like to be together. They're really social and they like to be in a group. And what they found is that when they took these animals that normally like to be social and isolated them, they do show these really stereotypical sickness behaviors. So there's a really great decrease in activity of these sick birds in isolation. But if you look at when they're in a group, there's no detectable difference between healthy or sick birds. So birds only exhibit sickness behavior when they're alone, which is interesting. So enter the zebra finch. Here I'm showing a female on the left, a male on the right. You can probably infer where they get their name from, that really beautiful striping that the male birds have. They also have those really gorgeous bright orange cheek patches, and zebra finches are native to Australia, but you can definitely have them as pets here. So you may have seen them in pet stores locally. They're opportunistic breeders, which means that they don't reproduce seasonally like a lot of birds. So they don't just reproduce in the spring. Whenever conditions are nice, so warm temperatures, lots of food, lots of water, they'll get together and produce offspring. They're also socially monogamous, so males and females pair together, and both sexes share parental care duties. So just as a little primer of stages of reproduction in birds, you may be familiar with this, but if we plot this on a timeline, the first thing that happens is birds will get together, meet, build a nest, and they'll mate. After that is some sort of incubation period where they have to sit on the eggs to keep them warm and foster development of those offspring in the eggs. And then at a certain point, they will hatch, cute little baby birds will be born, and then the parents have to work really hard to provide resources and food to those offspring that can't fend for themselves quite yet. And so the important thing here is that incubation is thought to have a lower energy demand for those parents compared to nestling provisioning, where the birds have to be really active, flying around, finding food, bringing it back to the nest. They often sacrifice eating themselves to be able to deliver food to their babies. And so this is an important concept going forward. Okay, so the main question here is, do breeding female birds behave like they're sick when they are given some sort of immune challenge? So do they exhibit those stereotypical sort of symptoms of being sick? Before I tell you the answer, I just wanted to include some pictures of how I actually find out. So this is an image of me working in our aviary. It's a large room, lots of birds are housed there. Again, zebra finches like to be social, they like to be in groups. An image of some of the nestlings that were hatched in some of the nest boxes of the birds that were breeding. And again, I'm going to mention this little trick that we use in research, which is to use this LPS injection, which tricks the birds into feeling sick and mounting an immune response, but it's only going to last for 24 hours. So it doesn't actually do any damage to their bodies, and they recover fully a whole day later. So what we did is we bred some birds, followed them, figured out where they were nesting, and then we only injected the female parents with this LPS to make them sick. And then we followed their behavior using video cameras and in-person live behavioral scoring. And we also collected blood samples from them so we could look at whether hormones were involved in controlling any of the behaviors that they might exhibit. So I'm going to show you some graphs now that you saw some pretty pictures, and I'll summarize the findings really concisely at the end. But if anyone's interested in data, I figured it would be nice to show some of the results. So we're going to be looking at the incubation time period first. So remember, this is less energy demanding than nestling provisioning, doesn't require a lot of work from our parents. And the focus here is to look at these light purple bars, which are the sick birds, compared to the darker purple bars, which are the healthy birds. But the main takeaway here is that basically in all cases, we do see this stereotypical symptom of sickness behavior. So declines in hops and flights, which is a measure of activity, increases in time spent resting when the female birds are sick, and decreases in body mass, which is a measure of their body condition. So it looks like in incubation, female birds do exhibit this sickness behavior pattern. When we look at the other time period, side by side, nestling provisioning, so again, this is more energy demanding, we basically see the same pattern. So again, declines in activity, increased time spent resting. This time we don't see a big change in body mass though. So the take home here is that yes, they do exhibit sickness behavior, but they only seem to suffer these body weight losses during incubation, not nestling provisioning, which is sort of interesting. Okay, so what about parental care duties? So if they're feeling pretty crappy, does this come at the expense of parental care? So here we're looking at the time spent in the nest on the left and the number of times the female visits the nest on the right. And here we don't see any differences during incubation. So there doesn't seem to be a compromise between taking care of yourself or taking care of your offspring during incubation. But in nestling provisioning, when that energy demand is really high, we see decreases in parental care when female birds are sick. So they actually are sacrificing their parental care duties when they're feeling under the weather. Okay. So lastly, I wanted to ask whether in these cases where we saw changes in parental care, do males step it up to compensate that parental care for their female partner? And the answer here is no. So you might expect that since these birds share parental care duties traditionally, that the male might just be able to take over for the female if she's off sitting on a branch somewhere, too tired to deliver food to her babies. But in fact, that's not what we see. So that could have really severe results for those offspring because they are getting decreased care from their mother and their father is not around to help pick up the slack. So this is a question that I'm asking now, which is how does something like single parenthood affect the way a sick female might prioritize themselves versus their offspring or their babies? And I don't have an answer yet, so that's coming soon. So this is basically happening this spring and summer, and hopefully by the fall, this question will round out this part of my work during my PhD at Berkeley. So you might be wondering now, this is all well and fine, but why study a non-human animal's response to an immune challenge? Why should we care about that? And there are a lot of reasons that I could list, but I tried to come up with the simplest answer, which is that I think it is really cool to understand that animal behavior is really flexible. And if we understand that flexibility, it allows us to better understand how battling an infection might impact reproduction and success of populations of animals in the wild. And importantly, this work shows that the timing or when an animal experiences some kind of challenge to their biology really influences how it might behaviorally respond. So with that, I just want to thank my advisor, Dr. George Bentley, my lab mates who are listed here, Popping the Science Bubble, for allowing me to share my time with them tonight. And definitely reach out to me. My contact information is there if you want to talk about birds or bats at more length. And if you are a Twitter user, you can also find me on Twitter. So thanks.
Great. Thank you so much, Mattina. We have a couple minutes before the next talk. If anyone wants to ask any questions in the chat or the Q&A, and it looks like we have one coming up. So one of our attendees asks, is vocalization an important part of incubation? Do you know if illness affects it as well?
What a great question. So vocalization in birds during incubation sometimes can be used as a cue to the partner to say, hey, I'm gonna leave the nest for a second. Can you come over and take over keeping the eggs warm? I didn't measure that for this project, but an undergraduate student actually that I mentor had a similar question. And so we did a side project where he made birds sick and then measured changes in song production or qualities of various bird calls at great detail. And I'm not an audiophile by any means, but it was really cool. And I don't have those results, but it's a great question.
If I could ask a question, obviously infections tend to get worse or harder to cope with seasonally, and probably like offspring survival differentiates seasonally. Do you see a difference in variance between these two conditions depending on like what opportunity birds took to mate, or does it seem to be pretty consistent?
So I think I can't really address like the time of year facet because I did everything at the same time of year and don't have any annual comparisons. But one thing that I think could certainly influence how they respond is sort of their starting condition. So birds that might be fatter and have more energy stores to start before I even did anything to them, they might not experience that energy limitation that I sort of mentioned. So there might not be a trade-off requirement, whereas birds that are smaller might actually have to sacrifice something.
Cool, thanks. And then we have another question also in the Q&A. How many years do birds live in the wild? Does it differ by species? So I think also more specifically about zebrafinch.
Yeah, so it definitely does vary by species. But zebra finches in the wild, I think the average lifespan is about 15 years, so pretty long. And in captivity, obviously, in our colony that we use for research, it can be way longer. So they do have pretty long lifespans.
All right, great. Thank you, Mattina. I think we'll also have a few minutes after the second talk and questions to kind of round out any further questions, and Mattina is more than welcome to monitor the chat, so everyone is free to ask questions continually. And I think Oksana is going to introduce our next speaker, Michael.
I think actually Jenna has the introduction for Michael, if that's right.
Yeah,
Yes.
I got it. Yep. Okay. So while Michael's getting set up, so Michael Manoharan-Valerio was born and raised in Puerto Rico and attained his undergraduate degree at the University of Puerto Rico at Ponce. He's currently a fifth-year graduate student at UC Berkeley in the Molecular and Cellular Biology program, and he spends most of his time listening to music and reading about history and sci-fi. Let's welcome Michael.
Hello, thank you for that introduction. Yeah, so my name is Michael, and I want to sort of guide everybody through the story of how your immune system is developed to differentiate between invading pathogens and its own cells. So I'm in the Roby lab at UC Berkeley, and we're really interested in these type of cells called T cells. But before we get to T cells, I kind of want to like build up to them, right? So if we take the blood of an adult human mammal, and you actually separate out between layers, you have all these different layers that come up. And I'm sure that we've already heard this term called white blood cells, right? So these white blood cells are these components of the blood, and usually it's this thick layer. And within these white blood cells, they are these cells called the lymphocytes. And in this group of lymphocytes are B cells, NK cells, and T cells. So we may be more familiar with B cells because there's been a lot of talk about antibodies recently, but it's actually kind of important to know that the function of B cells is very closely tied to that of T cells. So let's say we have immune response to something, right? So this is like a normal chart of how an immune response would go. So the immune response has two main portions. You have the innate immune response, which it's kind of interesting because Mattina talked about LPS, which usually is a stimulant for the innate immune response. So then you have what's called an adaptive immune response. So this is what happens 7 to 15 days after the initial exposure of the antigen. So this is where the T cells would come in, and subsequently the B cells too. And I think you can see a pretty good relation where you have a decrease of the viral load as these adaptive immune cells come up. So I like to think of it as like the elite squad. So if the first wave is going to take care of it, the second wave is for sure going to take care of it. Okay, so how do we actually know how T cells interact with their environment. So I'm just going to mention three main concepts to sort of get us oriented. So the first part is the receptor, the T cell receptor that is actually on the T cell. And this is the main way of how T cells interact with their environment. I'm going to talk about this in a little bit in the later on detail, because it has a lot of importance. The second factor is the peptide. So here I have this red squiggly line. And essentially what this peptide is it gives an example of how the cellular environment is on the cell that the T cells interacting with. And then the final component is this major histocompatibility complex. You can think of this as sort of a personalized docking station where you have where the peptide that gets the information of the cellular environment docks. And then when we combine these two components, we kind of get what we like to call or think about the check-in from the cell to the T cell. So with that in mind, let's try to go through a sort of a conversation of how a T cell in a normal healthy cell would go. So the T cell and the T cell receptor and essentially ask this question: are you my match? Okay. And if the healthy cell and the message that it's giving to the T cell says no, then there's no response. And this is usually because there is a piece of healthy cell on this message. So the T cell has no reason to go in and attack. So let's take another situation. So here again, the T cell asks, are you my match? It turns out that this message and this part of the peptide, and this peptide is the match. And it turns out that this is just a tiny piece of what a pathogen, of the pathogen that's invading this body. So in this case, what would happen is that an immune response would be generated against that pathogen, and then the pathogen gets cleared. So that's sort of like how generally this goes. So one of the big questions that T cell biologists had for many years was, how many T cell receptors can there be? Because an important part of this whole interaction is that this T cell receptor had matched with this specific portion of a pathogen. So how does that happen, and how does it know that it's a pathogen, it's not from itself? So a lot of years, a lot of research went into this. So I think we may have time. I kind of want to pose the question, if it's possible, if people can like give me a random number of how many T cell receptors you think are possible on the human genome. I can't really see if people wrote right. So can somebody help me out?
Yeah. So in the chat, we have a guess of 2,000. We have 100,000, 50 million. I think that's a trillion. Someone says infinity.
Okay. Okay. So it's not
millions
infinity.
and millions.
Well, millions and millions is pretty close. So it turns out if we just look at the possible arrangements of the genes, the number is approximately 10 to the 15th possible T cell receptors in the human genome. And just to give like a frame of reference, there are about 7.8, 10 to the 9 humans on Earth. So it's quite a bit. It's quite a bit of possibilities. But if we look at the blood of an adult human, turns out that there's actually less T cells than there are possible. There's actually around 2.5, 10 to the 7. So this is weird. This is a lot of these possible receptors are getting rejected or not being observed in the blood. So that comes up with this question: what happened to all of them, right? So this is something that my lab is really interested in, and essentially T cells have to go to T cell school. Okay, so the T cell school is located in the thymus, so it's this gland that sits right on top of our heart, and it's an organ that's really big and really active as we're young, but as we age, it starts to involute, and it's your potential immune responses or your potential generation of T cells also decreases. So that's why it's really good to like get exposed to stuff when you're young, because that's when it's the most active. Okay, so this is the T cell school of self-awareness. So within the thymus, let's say that we have these T cell precursors. This is just, it's a developing T cell, right? So this developing T cell has rearranged its T cell receptor. So that's this little part here. And as it's developing in the thymus, it needs to go through certain checkpoints just to make sure that it's developing the right way, right? So sort of like grades, you know, first grade, second grade, third grade. So then this like developing T cell interacts with a training cell. So this is a cell in the thymus. And this cell in the thymus shows them an example of a peptide from a healthy cell. So it gives them the example to test, okay, how are you going to react to this? And here's what we like to call the Goldilocks model, okay? So if you remember that story of the Goldilocks, you have a Goldilocks that walks into the house and just tries porridge or soup, depending on how you read it. And the first time Goldilocks tries, it's too hot, right? So this is sort of akin to what happens to the T cell. So if the T cell responds too strongly to the sign of a healthy cell, that T cell needs to get eliminated. And this is what we call negative selection. And the consequences of this not happening is that you would have a developed T cell that can mount a response to your own cells. And we don't really want our body attacking itself, right? The second porridge that Goldilocks tries is too cold, right? So this is also a problem. So, and in this case, the T cell can't even see or respond or doesn't even know that it's being shown something. It's completely not paying attention. And what happens is T cells, especially at this moment in their development, they're very finicky. They need to have signals. They need to have some sort of interaction. Like they need like some sort of affirmation that everything is going fine. If they don't receive that at all, then these cells are removed. So finally, after all that work, Goldilocks finds the right porridge. And in this right porridge, you get just enough of the signaling that your T cell gets that positive affirmation, everything is going well, you can move on. But not enough to really worry and go through the process of negative selection. So what happens, right, if T cells don't get negative selected? If you have T cells that are pathogenic at this point and escape, well, you have autoimmune diseases. And one such example is MS. So in multiple sclerosis, what happens is that you have these myelin sheaths that cover nerves, and the T cells are involved in that they've escaped the cycles, but they're still able to respond strongly to pieces of the myelin sheath. So in a healthy cell, that's just showing them, it's like, okay, this is part of me, everything's fine. And then the T cell is like, no, something's wrong. Something's wrong, and just like goes at it. And unfortunately, that has dire consequences. And a lot of autoimmune diseases like celiac disease follow this pattern when you have T cells that just overreact to something that should be normal. So what's my word? So it seems like it's just too simplistic, right? It's just like these three. Turns out there's another option. And these are what we call unconventional T cells. And they go through a type of selection that I like to call unconventional selection. So these are cells that are actually trained to respond strongly to proteins of self. But that doesn't really make sense after I just told you this whole story, right? Well, if we add a little bit of context, so it'll make more sense. So these are cells that are specifically trained and selected in the thymus to react strongly to peptides or signals from a host. But the peptides that they look at and recognize are peptides that shouldn't be in the MHC, in the message. So this is a signal of the host cell that by the mere fact that it is present there means that there's something wrong with the cell itself. And these unconventional cells, when they see this, they initiate a response. Okay, so question time. Why would you think that we would want cells that attack our own cells? Again, I can't really see so.
There is an answer in the chat, maybe cancer cells?
Very good. Yep, exactly. So this is kind of the three main reasons why we think that this might be a thing, right? So cancer immunotherapy. So if we think of a cancerous cell, right, it is a relatively normal cell, but it has a defect in its cell cycling, so it just replicates a lot, right? That itself is a very stressful process for the cell. So things that shouldn't be presented on the MHC will be, and that's exactly correct. Another possible reason is if you think back at the story with the pathogen, right? You would need a certain type of T-cell receptor for all these type of pathogens, right? Some pathogens are really good at evading the immune system. They're very good at hiding themselves. But because of the nature of the pathogens, they need to replicate and they need to put stress on the cell. And by adding that stress, they are messing things up inside the cell. And those stressful responses can lead to peptides that are from the healthy cell being put up on the MHC, which can lead to the immune response. So it's sort of like a plan B if the pathogen can escape the detection. And then the last one that could potentially be used is for therapy of autoimmune diseases. So if you have overactive cells, like in some cases of autoimmune diseases, they will also put up things that shouldn't be in the MHC. And the more that we sort of understand what leads to these unconventional T cells, the more that we can try to apply those cells to different therapeutics. So my big question is, what needs to happen in the science to break the rules of the conventions that we had so far that will lead to the development of T cells that recognize them? So after 5 years of nonstop research and just like blood, sweat, and tears in the lab, always looking fashionable, it really has been broken down with the cells that I work with just 2 factors. So it turns out that these T cells, these developing T cells, actually differ quite a bit. So the first thing is the cell that trains them is completely different than the cells that train the other T cells. And not only that, but the test that they're given is also different. So even though they are within the same school, it's kind of like they went like an AP route or something. They went through a whole different experience that prepared them for a whole different set of scenarios. And then that's sort of what we know so far. There's a lot of different unconventional T cells, and they've been recently more and more studied. So there is a lot to find out about this story. So I think I want to end that. I want to thank the Roby bunch and the Roby lab, especially my mentor here, Roby, the Costco lab, who works very closely with us on this project, and the Shastri lab, who Nilab Shastri, who has recently passed away, his lab were the ones that first described the cells that I studied. So we're really indebted to him. So with that, I would like to end, and I would take any questions. Should I stop sharing so I can see the questions? Or
I am happy to ask questions. You can keep sharing your screen or not as you see fit. Thank you so much. That's a super cool talk.
Thank you.
We have a couple questions already. So one is, how does the T-cell receptor locate its target peptide on the pathogen cell? Does the protein cover the surface of the entire pathogen or just a small portion?
Okay. So the way that works is there's another cell, so it's an indirect reaction. So there are other cells that are called phagocytic cells. So these are cells that eat up stuff, and then they show the pathogen, the piece of the pathogen, to the T cell. And then the T cell, depending on the flavor of T cell, will then inform B cells to produce antibodies, or depending on the pathogen, will go on and attack cells that are infected with a viral pathogen. So T cells won't directly go to the pathogen, but instead orchestrate the whole adaptive immune response in order to eliminate the pathogen.
Cool, great. And we also have another question, which first says, thank you for this talk. And also, what do you think would happen if these cells were knocked out?
Oh, so that's an interesting question. So there has been studies where you've knocked out some of these unconventional T cells. One of the most famous unconventionals would be what we call T regulatory cells. So these are cells that target autoreactive T cells, and you get massive autoimmunity. And these studies have been done in mice. There are other bunch of unconventional T cells that usually what happens is that there is an autoimmune or a complication in certain pathogenic situations. So, yeah.
Cool. And then Violet asks, are there still types of T cells to be discovered?
Most definitely. Most definitely. Even now, when I started my grad program, I came in with the mentality that there was a very fine line between adaptive and innate immune responses. And the more that I study it, the more it's getting blurred. Before, it used to be just as simple as CD4 and CD8 T cells, right? But now there's just a whole gamut of them. There's just so many. And I really think attribute that to, as our technology grows, we have more sensitive assays, and we can actually look at specific places and ask more directed questions that will lead. So yes, I think there is infinite possibilities.
Exciting. And I guess a good follow-up question to that is someone is asking, is the immune cell system the most complex system in the body?
That's not fair. I'm gonna say yes because loyalty, and two, I'm gonna say it's pretty complex. But I'm pretty sure that those are fighting words to another type of biologist. So
neuroscientists would probably duke it out with you.
Yeah, so I'm gonna stand my ground, but I am going to recognize that that may not be the case for everybody. In my taste, yes.
And then we have another question, which is it useful to look at T cell function for diagnosis of some sort of immune diseases?
Huh. I guess it seems like a simple question, but I don't really understand it. Because if we measure out the type of T cells in a disease, to like if we take out blood from a patient and see that there is more of a certain amount of T cells than others, would that be a diagnosis? If that's sort of the question, then yes, that would definitely be one way. Another way would be to look at the specificity of the T cells that are in the blood that are circulating, and that can definitely inform you of what is being actively fought. Yeah, if I'm understanding the question, then yes, it is worth it.
Great. I think that's currently all the questions we have on the panel. I don't know if there are any from the Facebook stream. It looks like no, but I really want to thank our speakers today. These have been super cool and super insightful talks and a lot of fun to listen to. Thank you guys so much for being here and for spending your time with us. So yeah, I think we have another seminar in a month. And this talk, if anyone missed any part of it or wants to renew it, we will be uploading it to the Berkeley Public Library's YouTube page, and then we are also making a specific Popping the Science Bubble page that should be more direct. And I think both of our speakers have added their contact info or ways to ask them more questions if you are ever so inclined. So thank you guys very much. I don't know if Kelsey, you want to say any final words about the library?
Yeah. Just thank you so much. These were great lectures tonight, and thank you, Popping the Science Bubble. And yes, look on YouTube for more videos. Everyone have a great night. Thank you so much.
Thank you, Michael and Mattina.
Bye. Thanks, everyone. Nice job, Michael.
Thank
Thank
you.
you.
Great talks.